Before I tell you the story about the work that's going on in our laboratory, I want to thank Bruce for giving me the chance to tell you about the work that my laboratory is doing. It's going to be a little bit different than what you've been hearing for the rest of the talks up till now. This is going to take a very basic turn, and we're going to also take it all the way to a nematode and how the nematodes shed light on the biology of this class of enzymes. And hopefully by the time we get to the very last few slides, you'll begin to appreciate how I believe, as well as many of my colleagues believe, that chromatin regulators, or these epigenetic modifiers, will be really important diagnostics as well as therapeutic targets. But before I get started, I really need to tell you about my core nucleosome, the core that actually makes all this happen. This has been driven by two very talented postdocs in the lab, Josh Black and Capucine Van Rijkum. It's been helped out by two very talented technicians, Emily, who was the very first one who took a bet on me and did really well, and then Andrew Allen, who has taken over the project and now has become a graduate student at HMS. So before we get started, for those of you that are less familiar with chromatin, what I'd like to do, or how the epigenetics may be playing a role, I want to kind of define it, because the next set of talks are going to deal with methylases, methylomes, and how this dynamic is relevant to cancer. So what I'd like to do is kind of set the stage for my colleagues that will follow just shortly thereafter. So what is chromatin? Chromatin is a highly ordered structure that contains both your DNA, your RNA, chromosomal, and histone proteins. And it's the concerted effort of the nucleic acids as well as the proteins that results in the packaging of your genome into your chromosomes. And it's this protein nucleic acid rich environment that results in very specific consequences. Structural ones that define, for example, what a chromosome looks like during mitosis. And then, as many of you think about, is gene control, regulating the appropriate spatial and temporal timing of gene expression, which gives rise to very specific cell fates, tissues, and an organism. And then, as many of you think about it, as you alter gene expression or as you're looking in tumors or cancer states, you're asking, are there key gene networks that are changing? So that could be changes in this RNA, DNA, protein environment, which results in inappropriate gene control, and in turn aberrant cells, and then the demise of the organism. But what I'm going to do today is try to paint a different picture for you. What I'd like to do is set the stage that chromatin is not just important in the context of gene control, but in fact, it's important in structural integrity or other very important nuclear processes like cell cycle or DNA replication. And that this opens a very unique door for therapeutic targeting or thinking about how tumors might be functioning. So the most fundamental subunit of the chromatin is the nucleosome. And here's a picture from Carolyn Luger's beautiful work in 1997, which she got the crystal for this nucleosome. And what I want to draw your attention to is that these long little tails sticking out of the DNA wrapped around your histones. These tails, what I would like you to do is I'd like you to think of them as antennae or whiskers, and that they're actually sensing or recognizing modulation in their microenvironment. So how might these tails or these that extend out, how might they pick up the signal? And they do this in the context of post-translation modifications on the histones. So the tails are very basic in nature. They carry a lot of lysines, as you can see here, and arginines. And because of this, they can be highly modified. They can be acetylated, which many of you know through HDAC inhibitor drugs and HATs and so on and so forth. They can be methylated and this is often thought of in heterochromatin or repression of gene control. And then you can also have a series of other modifications like phosphorylation, ubiquitination, and so on. And it's thought that the combined effect of these modifications can then establish very specific consequence or help aid in certain nuclear events. So what my lab is interested in doing is understanding how this microenvironment bridges two particular areas. And when you first glance at this, you're going to think he needs the broad to do this work. But what we're trying to do is use the fundamental insights of very specific enzyme pathways to kind of bridge two concepts. One, how is it that you maintain a proper cell fate decision by regulating both the gene control, the natural endogenous transcriptome, and the major component of the genome, which is this non-coding portion or this region that's being expressed? How do you control these two events in the context of a cell that needs to undergo a division, as well as maintain its chromosome stability and structure without activating a response or DNA damage pathway? Said differently, if you misregulate these, then how are there any kind of bridge between the two events? And so what we actually believe is that methylation actually serves. Histone methylation can serve as a wonderful bridge between these two dynamic parts of biology and may actually be fundamental to what we see in the context of chromatin regulators. So what makes lysine methylation on histone so unique? I think of it as a multi-layered cake. You have 3 different layers to it. You have the type of lysine, the specific residue. You're going to hear, in my case, of lysine 9 trimethylation or lysine methylation. You're going to hear about lysine 79 and a variety of other modifications. Each residue has its own kind of related consequence. Then you have the degree of methylation, and this is just due to the chemistry of the site. Lysine has an amine, which can be mono, di, or trimethylated. And each one of these has a different consequence or is associated with a different event. So you have now 2 levels that make it different. And the third one is, where are these physically placed in the genome? And you're going to hear a great deal of this from Brad Bernstein's work, which has really pioneered the understanding of how specific cells have specific states. So I'm going to use a very, very simplistic cartoon to illustrate the point. And this oversimplifies a lot of data, but it just highlights a couple key attributes. The first one is the sense that you'll notice at a transcription start site, you have a peak of K4 trimethylation. Notice I'm not saying monomethylation. In fact, monomethylation on the same residue is found in enhancer elements. At the same time, if you methylate to a trimethylated state, a residue only 5 amino acids away, it has a very strong repressive function. And this has been associated with gene repression. At the same time, if you move this nucleosome, this modified nucleosome over, it's often linked to actively induced genes. So what this does is this highlights it's not just it's the degree, but also placement becomes critical. So therefore, the cell needs cells or needs enzymes that can help modulate this different degree. But as I indicated in the very beginning, the regulation of these modification states is not just important in the context of gene expression. So as one is sitting and thinking about epigenetic regulators, one needs to consider the bigger picture. How does it actually impact the nucleus and the organization? And what do I mean by that? Well, in the case of euchromatin, which is your more open region of the genome, we know that this replicates earlier. It can have kind of a diverse pattern. You can have early replication or late, but we know the region that's highly compacted, highly organized, and carrying great deal or high levels of K9 trimethylation and increased heterochromatin proteins, that you get a very tight, compacted structure which replicates late. And it also is very key to when you look at a chromosome, because those regions that are highly compacted are your telomeres and your centromeric regions. They're enriched in this modification. Therefore, if you mutate, delete, or alter the enzymes important for this, you can have very distinct problems in genome stability, especially in these targeted regions. Now, this suggests a structural function, but what I'd like to do is highlight that now if you look at an interface nucleus, this becomes just as important. You can see that the heterochromatin resides typically in the periphery, and this is this dense structure here. You can see that the euchromatin here is this light gray region. So what this suggests is that the heterochromatin formation, the methylation state, is not just important in the context of genes, on or off, not just important in the context of your mitotic chromosomes, but has a very important role in potentially establishing your zip code or where you're localized within the nucleus. One of the most beautiful pieces of work, I think, that illustrates this point is from Dave Gilbert's lab, where they took ES cells, they differentiated them into a fibroblast, and they looked, for example, at heterochromatin markers, and they looked at replication, and they found that there was a very big difference. However, upon reprogramming the fibroblast back to an induced pluripotent stem cell, they found that it looked more like an ES cell. But then when you promoted it to differentiate, it now had the same pattern as the final fibroblast. So what this says is that there's an inherent mechanism that's allowing these more global events, this global structural integrity to be maintained. And so what we're interested in is looking at the enzymes that are involved in this process and how might they be modulating this, and then trying to take this from a very basic level into potentially either clinical diagnostics or asking, can we use this molecular insight about these enzymes to be great therapeutic targets? The first demethylase that was discovered, and we're going to talk about the demethylases in this context. You're going to hear more about methylases in a few minutes. There are 2 classes of your histone demethylases. There's your LSD or your FAD-dependent enzymes, which were discovered by Yang Shi at the medical school as a postdoc and his team. Then we subsequently went on to discover, along with many other laboratories, the Jumanji-containing oxidases, which are tri- and di-demethylases. Now, what makes the Jumanjis very interesting, and this chemistry will be, I think, intriguing to many of you, those of you especially who think about chemistry or compounds, is that they require three key features. They require molecular oxygen, alpha-ketoglutarate, and iron 2. And I'm showing you here the snapshot of the crystal structure that we solved a few years back, and it coordinates your alpha-ketoglutarate and your oxygen. Here's your position methyl residue. And as you basically get a shifting of electron bonds, you basically generate this important species here, which is an oxoferral species, highly, highly reactive, basically abstracts a hydrogen, creating a hydroxyl group, which then is put onto this carbon. And this creates a carbonyl group, and this carbonyl group is a fantastic leaving group. So it spontaneously demethylates or creates formaldehyde. And as you might realize, a lot of these are metabolic or oxidoreducto sensors. So this may be very important when you start thinking about tumor burden and vascularization and so on and so forth. So the family of enzymes that my laboratory is extremely interested in are the Jumanji, or the KDM4 family, or the JMJD2A family of enzymes. There are 4 invertebrates. There's 2A, 2C, 2B, and 2D. Then we're very fortunate that in C. elegans there's 1 functional homolog. So what we set out to do is really interrogate their biology and understand how these enzymes might be functioning. They're regulating lysine 9, and they've also been implicated in regulating lysine 36. So like any other enzyme in the world, you need to know what they do biologically. So what we've done is we use the C. elegans model, which is this really powerful genetic organism, to really fish out the human biology. We look for conserved function. Best experiment yet was my daughter catching the fish. She didn't use the worm. They're only 1 millimeter in size. So basically, the beauty of this organism is as follows. They have invariant cell division and growth, which becomes very powerful for us. They have a complete cell-fate lineage map. With their transparent, so their ability to watch cells or to do labeling becomes critical. And the third one is the power of the forward and reverse genetics. And this work was both carried out by these 2 talented individuals. So the story I'm going to tell you about, and this is just it in a nutshell, is that the demethylase actually regulates your nice lysine 9 dynamic and the recruitment of a very specific heterochromatin protein, and that this is critical in regulating how fast one progresses through S phase. How did we get there? We got there because we used the worm model. And what we found in the C. elegans model was that when we knocked out the demethylase by RNAi or isolating a knockout, we got increased apoptosis in a very specific region of the germline. This was indicative of a DNA damage or an apoptotic checkpoint mechanism in worms. Based on all the mapping and all the things that have been done, we could pull this information. It wasn't physiologic apoptosis because no apoptosis occurred anywhere else. This is indicative of 2 effects, DNA damage and p53 activation. So we looked for genome instability or damage, and we found an increase in the mutant. We then knocked out p53, and we could rescue the apoptosis. So what we then wanted to know is, what is the pathway? What is reading this damage or this change here and then giving you a readout down here? So we did a series of epistasis experiments, knocking out kinases and asking what would happen. And what you can see is we identified ATR or ATL1, the homolog, in worms to rescue. So if you knock out ATM1, you don't rescue the increased apoptosis. However, if you knock out ATR, the kinase involved in replication completely rescue the phenotype. So this was our first suggestion that the enzyme may be modulating replication. We then went on and then took advantage of the organism, and we did live imaging or pulse chase experiments in the germline where you put in 1 fluor to look at replication, you wait a period of time, you pulse in another fluor, and then you look at the readout. And so if in fact replication is impaired or slower, 2 things should happen. You should have more nuclei in the stem cell zone that have red and green label. More importantly, you should have a co-label, a yellow region. Take-home point is you get more red and green nuclei, and then you only see in the mutant these yellow regions, this overlap. So this was very strongly suggesting that there was an impaired replication. As I said, we used the worm to fish out human biology, and then we hope to take this back into cancer models, tumor models, to better understand maybe how we could look at the levels of these enzymes and then think about how we might use either classic or new therapies. And this work was then carried out by these 2 talented postdocs, Josh Black and Capucine. First thing they noticed was that one of the human homologs had a very unique distribution in cell cycle. It was high in G1S, came down during S, and was almost obsolete in G2M. And this was very intriguing to us because we noticed that it didn't have a change at the transcript level. Upon further examination, we found that it was regulated by the proteasome. Capucine then went on and elegantly demonstrated that the regulation through cell cycle was through the SCF complex, in particular FBOX4. So this identified an enzyme that was being highly modulated during S phase going into G2M. So this begged the question, is this the enzyme that may be the counterpart or playing the role of the worm homolog? So we look closer, we overexpress the demethylase. It's very important to realize we're only getting about a half to twofold overexpression of this enzyme. And then we made catalytic mutations. Take-home point is they progress faster through S phase, so we arrest them in the cortisol and release. You get more here at 12 hours compared to control or the corresponding catalytic mutation. And this is quantifiable. Josh really wanted to know why. Why is it that we're impairing replication in ACL again, or why is it that we're affecting S phase progression? So then what he did is he asked the question, are you getting more initiation events, more replication forks? And in fact, we took advantage of this beautiful antibody, the BRDU antibody. It only recognizes single-strand DNA. So if you use an antibody against it, you only see single strand. If you denature, then you see total DNA content. So replication forks, those regions are going to have single strand. You'll be able to set up a ratio. And what you can see from this is that you're getting more when you overexpress the enzyme as you proceed through S phase, suggesting that there's more replication forks, more replication initiation events. We then really wanted to look at this at a specific locus and ask, are we in fact seeing changes in replication at specific loci? Is the demethylase actually specifically regulating certain parts of the genome in this process? So we took advantage of the BRDU again and chipped it at these particular regions. We looked at satellites, which you'll hear about more probably from David Ting and so on, in the sense that satellites are late replicating and they're canine enriched. Then we looked at alus, which are kind of a different species in the sense they're early replicating and canine poor. And what we actually found was that only the satellite region, as well as a few other regions like centromeres, actually had an increased BRDU incorporation in a catalytic-dependent fashion, whereas alus had no effect at all. This suggests that the demethylase has the ability to modulate very specific regions, the replication of very specific sites. We then carried this out genome-wide, and we asked, in the cells where you have increased 2A levels, and you look at 2A occupancy, which is about 2,400 positions, is there more replication occurring around or at the peak of binding? And so when you look at this, you see that as you increase 2A levels, you get a very substantial increase in the amount of BRDU that surrounds or is at the focal point of binding. And if you take the same number of sites and randomize them across the genome, it's flat. So this suggests that this is actually an event that's occurring genome-wide, but in very specific locations. Josh then asked a very important question. If you're changing S-phase progression, making it happen faster, and you're modulating BRDU, then you must be regulating timing of the genome. Certain regions' timing of replication may become very fundamental and change. And if you're thinking about using drugs to target a cell that's dividing, this becomes very important because the satellite regions or these repetitive regions, you want to gain access to them because if you can increase how open they are or you can take advantage of this, you can promote increased damage events. So what we found is that the demethylase actually resulted in an earlier replication timing. So what we do is we take cells that are actively and happy, no drugs, we fractionate them as they go through cell cycle, then we chip them and we look at very specific loci. Now we're doing genome-wide approaches using this, and you can see that you shift from late replication or mid to late with a satellite to early, and it's very specific because we don't see it at other regions of the genome. Once again, suggesting that demethylase has the ability to modulate very specific regions as you're proceeding through S phase. We know it's a target. The demethylase actually occupies that physical space. And then Capucine did a very important experiment where she asked are these enzymes coming and going from the chromatin? And has this become very important? Because you might think as you're strategically targeting certain cancer types, dividing versus non-dividing, slower cells versus others, this may be very important. And what she thought was, at early S, maybe what you'd see is an accumulation of the demethylase. And as you proceed later at certain sites, it would come off. And that's exactly what we saw. So what you see here is this is control. This is just to show we get overexpression. But take-home point is the pattern's the same for control. As you're proceeding through S phase, you get an accumulation at the satellite region, which is then proceeded to it going down to a basal level, but never void. kind of like what you see with pioneer transcription factors. They go to very specific sites. They never leave them. Then you get an accumulation, which results in an event. Josh then wanted to ask the question, if you're seeing all these changes, what might be regulating this? I've kept alluding to how open the chromatin is and how open the genome is. So he took advantage of the micrococcal nuclease experiment, which cleaves around your nucleosomes. The more compacted, the less access. So this will tell you if the genome is in fact more open, which can give you some ideas as to whether it's a good target. So what we did is we did the MNase experiment, and we found that there is in fact more mononucleosomes compared to either catalytic mutation or your control. So this is a mononucleosome, dinucleosome, tri, tetra, so on. But the real proof is if you see the region of interest is actually more open. And in fact, here's your satellite. This is a southern blot, and you can actually see that it's more open. Josh wasn't satisfied with this. He went on and asked, if our hypothesis is true, that you're changing distinct regions, you should see it opening as the cell is proceeding through cell cycle. And it should change. The timing of opening should change. And that's exactly what we see. So this was arrested and released cells. At 2.5 hours, we see that there's a peak opening of this region. And you see that 5 hours, they're more similar to one another. So this just suggests that the demethylase is regulating how open very specific regions of the genome are during DNA replication. How might that be? Logical approach is to ask HP1 proteins, because HP1s are these heterochromatin proteins which bind or read canine methylation. They condense and consolidate a region, so they would make be a logical candidate. And many of you have probably heard in textbooks heterochromatin is heterochromatin. The truth of the matter is our data began to show us a very important insight into heterochromatin formation. So here's a quantification of our facts. Here's a control cell, early, late at 12 hours. You see a shift. It's more late in the 2A overexpressing cells. We then overexpress the HP1 proteins to see if we could counterbalance JMJD2A. And what do you see is that HP1 gamma is the only one that's able to actually antagonize the demethylase. Alpha and beta, which have been thought to be the critical regulators of those chromatin structures and replication, do not. And we also then went on to demonstrate that it was through the chromoshadow domain, which is an oligomerization domain, and through the binding module, which recognizes the methylation state, the chromodomain. So this really establishes that you need very specific enzymes to counterbalance very specific readers or things that recognize those distinct regions throughout the genome. So we found that it actually rescued replication at these sites. And then what was really important is Capucine then went on to demonstrate that in a temporal fashion, you reduce canine trimethylation at satellite 2, and at the exact same time is when you achieve the lowest level of that heterochromatin protein recruitment to this position. So what this did is this is establishing a model by which a demethylase, by regulating this level through S phase, you're modulating the distribution of heterochromatin proteins, the initiation events that occur, and also potentially how fast the polymerase is elongating. We have replication sequencing data that suggests it also influences this process, where the process is determining if it's an equal split or if it's favored in one way or the other. And we actually were able to observe all this because we could then go back in worms, and we actually identified a very specific heterochromatin protein that actually antagonized the demethylase. So this is conserved from worm all the way to human. But then everybody at the end of the day wants to know, what does this really mean? How does this impact potentially what we're going to ask a question about clinically? If we're a pharmaceutical company, we're thinking about targeting, how is this relevant? What might be a good angle on this? So by taking these very basic and molecular approaches, we are generating what you'd classify as like a fingerprint about the molecular role. Is it gene expression or is it chromatin modulation in cell cycle? By looking at this, we can ask various questions. Our model would suggest that potentially having increased levels of this enzyme for family members of this enzyme, you might change their sensitivity to drugs that target this pathway. So that's exactly what we found. In fact, if we use replication type drugs, I'm showing you an example for HU, but what you find is that if you RNAi the human gene, you see increased sensitivity to replication drugs. If you use a catalytic mutant, you get increased sensitivity. Now think about it. If you're targeting the enzyme, you may create an inactive enzyme. So maybe this is acting like a dominant negative. On the other side of it, increased expression, only about a half to twofold, you get faster recovery from treatment. And with that, I'd like to thank the group that did the work, my collaborators, and of the funding sources. Thanks.
I want to ask you a simple question, Jonathan. My concept of methylation and demethylation, the effects on DNA were that methylation unwinds or decreases the compaction, I should say, and makes it more accessible for transcription and replication. Is that not the case with it? It would seem like the opposite here.
So the question is about how the methylation dynamic. So the idea is that certain methylation events are linked to more active open regions. So if you have things that are associated with active transcription like K4 trimethylation, that would be linked to more active euchromatic regions. Whereas the types of methylation and the things that we're talking about are more in the regions that are less accessible. They're thought to be more repressive, more condensed. So as you relieve that kind of condensation, you now open it up. And by opening it, we think what's happening is you're allowing more of your initiation machinery for replication to gain access.
I see. Okay, that helps.
Yes.
Yeah, Jonathan, very nice talk. Just wondering, you show beautiful dynamics with the demethylase. Do you ever look at the methylase? Do they have similar dynamic kind of during the cell cycle? And do you observe antagonism in general between the methylase and the demethylase?
Yeah, so this is actually a great question. So what we've done is, since C. elegans provide us a very powerful genetic model as far as being able to analyze this, both just simple, like a simple screen, we've actually done this, and we started looking to see if there's a clear antagonism. It's a little complicated. It looks like there's actually a couple methylases that may be actually counterbalancing this particular enzyme. And so now what we're doing is we're wanting to go and look in the context of cell cycle, are they regulated? And what's beginning to emerge in the field is that through the microcontrol, through post-translation modifications, many of them are actually regulated. A well-known one that a lot of people forget about is MLL. MLL is highly regulated during cell cycle. So we don't think it's just this demethylase. We actually think it's actually a process that's occurring for many of them. And potentially this regulation could be key to understanding how these enzymes exert their most important function on the cell or the type of tissue or so on. So we're looking at it now. I don't have a clear answer as to which one, but we're testing it.
Other questions?
All right.
Jonathan, one other thing. You mentioned, the potential of inhibitors.
Yes.
How do you attack an enzyme like this with an inhibitor? What would be the sort of the chemistry that you would go after?
So this is a huge area of investigation. Many companies, biotechs, academic scientists. So far, what's out there as far as inhibitors, many of them are either mimicking the alpha-ketoglutarate,
Right.
or many of them are actually targeting the iron center. So you might envision that those particular regions would be great targets. The only thing you have to be cautious with in that is if you target them, are you going to affect the other alpha-ketoglutarate or iron-dependent enzymes, which are about 2,500 in the genome? But I will say something that I didn't state probably clear enough, is that methylases and demethylases are exquisitely specific. They regulate very specific degrees of methylation and very specific sites. So if you can modify a very specific chemical structure, I believe based on the crystals we have, the co-crystals we have for many of these proteins right now, is that you can actually achieve a high level of specificity. Yeah. So I think it's
similar
possible.
to the tyrosine kinase
Yes.
inhibitors.
Yep.
Even though they look, the general mechanism is similar. The specifics of substrate and protein are very different.
Yep. Thanks.
Well, very good. Okay.
Thanks.